Perfect. Thank you, Shaylee. Yeah, just an introduction. I'm Brian Weed. I'm the VP of Sales here at CCS. My background is that I've been in the life science sector for 15 plus years now, starting out in doing the biodecontamination across North and South America, to being here at CCS for 7+ years. Got my start over at Villanova University doing a mechanical engineering degree. So I'll then go and introduce Ziva Microwright, who's the CEO, or Ziva Abraham of Microwright. Go ahead, Ziva.
I'm the president and founder of Microwright. By education, I am a microbiologist, but I have a lot of experience in clinical, industrial, and pharmaceutical microbiology. My focus is mainly contamination control because of my medical and clinical background. I focus on contamination control very seriously. It's a science by itself, and we all know that in this day and age of increasing immune compromised patients, contamination control is very critical. And we will be looking at cleaning and disinfection, all from a contamination control perspective and patient's perspective.
Great, thanks, Eva. I think just as a way of just a kind of reminder, what we're going to be covering in this is just really the science behind the cleaning, just as Ziva has said, and where we're going to talk through just some of the upcoming changes that we're coming out in the EU Annex 1, along with other impacts that we're seeing in the cleaning and disinfectant studies, and really kind of going throughout on what practices that we've seen that really have led to contamination for the industries that we service here.
So to understand cleaning, the first thing you have to understand is the capabilities of disinfectants. You do not choose willy-nilly your disinfectants because you think that is going to work. You have to understand what each disinfectant does or does not do, its compatibility issues with the hard surfaces in the clean room, its capability of leaving residue and why remove residue, why breaking surface tension is so important. And that's where your supplies come into play. You cannot apply and soak the facility because you all know what is behind the walls. You have your wall materials, which already has sawdust, glue, cement, paper, and all that is already laden with mold. So people who do excessive cleaning end up with more mold contaminations. And the most important is, can you reach the nooks and crannies where the organisms could be hiding, and then if you have good temperature and moisture, they may start growing and you can have major contamination issues.
Yeah, Ziva, I mean, the one that we talked on there and leading up to this, I think the big thing that we talked about was the applying without soaking, and that there really is a true form behind that, because as we'll see later in the presentation, you don't want to oversaturate it, and even the different surfaces and how they look differently on a microscopic level, I think are really a key thing to understand when we're talking about the science behind it. I mean, I know when you go through and you do talk about some of the disinfectants, and I know this was the facts you really wanted to kind of hit.
Perfect. So let's understand the disinfectants. So in general, in the industry, we use phenolic compounds, and you can call it Vesphene, LPHSE, deconfene, and anything else. These are general purpose disinfectants. I want you to understand very quickly how these disinfectants are categorized by the FDA and the EPA. They're categorized in three distinct categories. One is the high-level disinfectants, which are sporicidal in nature. Then you have the general purpose disinfectants, which are disinfectants but not sporicides. And the third is the sanitizer, which what I call them as wishy-washy disinfectants. That is your IPA, which hardly kills much, right? So phenolics and quads that we use are general purpose disinfectants. But why do we use them? We use them because they have a surfactant in them, and you want to break the surface tension when you use a disinfectant. Just example, right? When you wash your hands just with water or you go into the shower, there is beading, correct? That beading does not give you good coverage, so you need soap, so the disinfectant gives good coverage on the surfaces. But both phenolics and quads leave residue, right? And there's a big focus, and we'll talk about Annex 1 2020 version, why there's a focus on residue. First, it doesn't look very good and not pleasing to the eye, but it also can become a source of contamination. The other thing I want you to, from my experience, I wrote the first chapter for USP on disinfectant qualification. That was the draft. I drafted it, and Dr. Roger Daba then put it into USP on disinfectant qualification. Um, I have done many studies, and have been involved in recalls. Phenolics are good. They kill all vegetative bacteria, some spores, even if there is a fungicidal claim. Quads have the same kind of claim, but quads are prone to growth of microbial contamination, especially gram negatives. And I've been involved in recalls. That tells you that you have to manage your disinfectants well. Then comes bleach. Bleach is still the gold standard, right? Number one, it's inexpensive, but I want you to understand that bleach is as good as available chlorine in the bleach. And once you open a bleach canister, the available chlorine drops. And that is why I encourage people to always know how many parts per million you have of available bleach. You use that for your disinfectant qualification study and check it every time you make a dilution. Then you have hydrogen peroxide. Hydrogen peroxide is the safest disinfectant. If we didn't have hydrogen peroxide in our saliva, we wouldn't have our teeth. It's used in food industry to clean all the canisters, but I want you to know at 3% it kills vegetative bacteria, and at 6% it just starts getting sporicidal. Now we use it at 31% for VHP. 10% is pretty good, but as a sporicidal agent, but commercially it is not available. Next slide, please. Then you have the peracetic acid chemistries, that is acetic acid and hydrogen peroxide, which is your Decon Spore or Spore Cleanse, whatever company you're buying it from. This is a great oxidizer. It's a good disinfectant. It's sporicidal agent. And I know a lot of people don't like it because of the vinegar smell. Well, what is wrong in being in a Vlasic pickle jar if PAA chemistry is doing its job? It is also used sometimes when you don't have a CIP cycle and some for your tanks. People use and hold it for 24 hours. The vapor itself kills a lot of bacteria. Aldehydes, I would not encourage anybody to use aldehydes ever because they are greatly carcinogenic. Many years back, I used to do disinfectant qualification studies. I thought I was invincible till I did a disinfectant qualification study with glutaraldehyde, and I will never come close to it. Alcohols are, as I said, wishy-washy disinfectants. The EPA and the FDA says it is an agent that reduces the number of organisms as accepted by the public health ordinance. I haven't seen that public health ordinance. If you find it, please send it my way. kills human-borne contaminants like gram-positive cocci very easily, but gram-negative and bacteria and mold grow happily in alcohol. They grow very well. They drink alcohol. That is why, microorganisms are our ancestors. Now you see why we hit the bar every Friday. If they like alcohol, we like it too. Uh, but the 30% is very important of water, because that allows the alcohol to go through the cell wall barrier and go into the cell contents and disrupt the cell function. So 80% or 90% does not work. It sits around the cell. The cell feels very happy after after a couple of margaritas, but it's not dead.
So, Ziva, I think one of the things that you and I both had talked through was that the case study that you had seen for the mold growth inside of the IPA storage.
Oh, yes.
Did you want to tell that story?
Oh, I have tons of stories. I will give you a medical device story very quickly. This was a medical device company, and I was much younger. I thought I was Sherlock Holmes, and I would go to the client. Now I don't. I've gained enough experience to tell them this is what is wrong. They called me and they said they don't see bioburden, but they're saying endotoxin. And I said, okay, great. So I went there and I started looking for water source. There was no water source till I was watching the operators, the assemblers assembling the stents. And I looked at the bottle of alcohol and it was murky. So I said, why is it murky? Do you buy it bulk? They said, yeah, we have a bulk with a tap and we fill our spray bottles from there. I took a sample and it was full of gram-negative bacteria. Another stem cell company I went to, and this was many years back, when I started walking through the labs, all their alcohol bottles has nice floating fuzz in it. So alcohol, I want you to understand that, and I'll explain to you why you don't take label claim at its face value. And it's very important that you understand the disinfectants. If you're doing a disinfectant qualification study, you do it right. So alcohol will not kill gram negatives, many of the gram negatives and many of the fungi. And we will talk more about the label claim. So these are your 4 organism types, right? You have the gram-positive cocci, which are very zen. If there is no food, they will lay down and die. Then you have the gram-positive rods that are yoga masters. They slow down their metabolism and heart rate, form a spore, and can stay there for 100 years till conditions are good. These are just like the gram-positive cocci, whereas the gram-negative cocci are very spiteful. You try to kill them, and what do they release? Endotoxin. And this is exactly what happened at the medical device company. The gram negatives died, but they spewed endotoxin, and they were failing for endotoxin. Next slide, please. It's very interesting that bacteria are 4 types and mold are 4 types. I want you to understand mold. You won't become a mycologist by looking at one slide, but you'll get a very good understanding. There are 4 types of mold: zygomycota, ascomycota, basidiomycota, and deuteromycota. Okay. Zygomycota, and I will be a little politically incorrect. I'm going to say they're very protective about the babies. You see, they protect the baby in a single layer of protection. Right? They even sometimes have a cushion inside so that the babies don't have the shaken baby syndrome. When these spores are mature, they will burst open and release millions of spores. But these are very interesting mold because they don't need a partner to make these spores if the food is ample. But once food becomes difficult, what do? They take the mycelia and they make it into sexual organs called gametangia, and they make spores which are tough to kill. This you might be able to kill with your phenolics, quartz, and sometimes even IPA. These may be very difficult to kill with your regular phenolics or quartz, and sometimes even sporicidal agents. Now, these guys, Ascomycota, are even more protective. They protect the spores in two layers of protection. They have a fortress outside. Inside the fortress, there are sacs, and inside the sacs, there are spores. Now go and figure out if the disinfectants are going to go through this fortress into the sacs and kill the spores. They don't. This is why contamination control is very important. You understand the disinfectants, but you also prevent these from coming in. Now, if you're seeing portabellas and crimes and shiitake mushrooms growing in your clean rooms and smurfs running around, please shut down your clean room. The fourth type is Deuteromycota. This is my generation. I am just joking. It's a hippie generation. Here, they don't protect the spores. The spores mature, they are released into the environment immediately. Now, this is what we see most, right? And everybody will agree. We see a lot of penicillium, we see a lot of aspergillus, Beauveria, Pacillomyces. And among them are the colored Deuteromycota. Why am I talking about this mold? I want you to understand, even though we are talking about cleaning, I told you contamination control is cleaning is an important part, but it is not the overall contamination control. These guys, the colored Deuteromycota and the Ascomycota, are the engineering squad of the mold army. When a tree falls, these guys are the first one that will break down the cellulose, the wood, into simple sugars, and these guys will come, these guys, bacteria, and make it into soil. What does that tell you? You should have a gratitude prayer for mold every day. Without mold, we won't have soil. You won't have your steak and your bread. Let's move on, please. But you don't want them in the clean room. So, let's say Brian decides to make a new disinfectant. If he's making a disinfectant for reusable medical devices, for decontaminating reusable medical devices, he goes to the FDA. Okay? It's treated like a medical device. If he says, I want it for clean room disinfection, he goes to the EPA. And EPA tells him, Brian, you have to do simulated use testing, actual use testing, biocompatibility, toxicity, material compatibility, and chemical indicator. This is something that is tested for bleach and PAA chemistries, but not many people know about it. So what happens is he has to do this simulated actual use testing using different organisms. But if you look at, now that you know a little bit about mold, if you look at the organisms that are tested for mold, that is your fungicidal activity, they are the Deuteromycota, the weak ones. And then you struggle with the strong ones and say, we can't kill it. They were not tested for fungicidal activity. Please do not blame the manufacturers. It's not their fault. It is EPA's fault and AOAC. What is AOAC? AOAC is Association of Analytical Chemists. Being a microbiologist and an Israeli, I will say it with total chutzpah, chemists don't understand microbiology. Otherwise, they would have chosen different organisms. And all the organisms tested for bactericidal, fungicidal, or sporicidal activity are weak organisms. This is why the regulators want you to do this disinfectant qualification study. Next slide, please. The other thing I want you to understand, there are interactions. Cellulose. How often, Brian, you've gone to a facility and called me and said, Ziva, they're using cotton string mops. I have a horror study for you here. I was called, this is 20 years back when stem cells was a new thing. They had mold everywhere. I went there and I saw a chemistry lab turn into like two little, put 4 HEPA or 6 HEPAs, put curtain and called it a clean room, right? As soon as I walk into this lab, I see a Home Depot bucket with a cotton string mop in it. That is how they clean. Now, bleach, and they were cleaning bleach. Two things, cotton string mops, cellulose. You saw fungi break cellulose. They are happy with cellulose. Bleach loses its activity over time, especially with exposure. Plus, bleach and cellulose don't jive. Cellulose reduces the activity of bleach. The same happens with bleach and quaternary ammonium compound interaction. This was a company on the East Coast. They used to clean with quaternary ammonium compound and put bleach. What happened? They had all kinds of contamination. They didn't know why. When I walked there, they had shoe covers and their whole floor looked blue. Why? Because the floor was sticky and your shoe covers were, shedding, right? Sticking to it. There was, because of the interaction, they did nothing actually. There was no decontamination or disinfection. Next, please.
Yeah, Ziv, I think to get back to your point on the cellulose and the bleach, there is a case study that didn't make this webinar that hit on that exactly. It was the same type of aspect that they were using the wrong type of mop heads, string mop heads, along with cellulose breaking down and all the different problems that went along with that. So you are correct. It is all too often we're seeing some of those different incompatibilities that you're looking in that side.
Exactly. So, for disinfection, the chemical kill, it's not magic. Just the chemicals will not do the magic. You need physical removal. If you are using incorrect supplies, right? And your supplies are don't break surface tension, right? And I will tell you why. Because I'm going to make another point. We talked about quartz and phenolics having surfactants. Bleach, peracetic acid chemistries, and IPA or hydrogen peroxide do not have surfactant. So if you're cleaning with those disinfectants, they are not going to break surface tension on the surfaces. And the smoother the surface, the more surface tension you have. So your supply should be those that break surface tension. And there are good supplies out there that are known to break surface tension. And then adequate application of disinfectant without soaking, ability of the supplies to cover all surfaces, uh, physical removal. Supply shouldn't be spreading contamination. They should be removing and holding contamination, right? And good coverage and maximum contact time. And we can talk about contact time when we go to Annex 1.
Yeah, I think the other one that Ziva, when you talked about the physical removal, I know that some of the different things that we've seen, honestly, that we've seen overall is a little bit in the industry trend that they're using a peracetic acid compound, then doing a physical removal using a detergent rather than the other way around. Correct? Isn't that what you're saying?
Exactly. You do the detergent to remove dirt and debris. So, it's been misinterpreted for years, okay? And I am going to tell you something. Disinfectants don't stop working after 10-minute contact time. The 10-minute contact time is the first contact time where during AOAC testing, they get no growth in the tubes, right? Does that mean that after 10 minutes, the disinfectants stop working and the microorganisms in the tube will resurrect? No. And this is a big misunderstanding in the industry, and we have to keep this point straight and make people understand. As you said, You remove the dirt and debris and let the disinfectant sit and do its job. Why do you kill it in its infancy? Just started working. Let it work. And there are studies that you can do to show that disinfectants actually last for 5-6 hours, depending on the nature of the surface.
I think that's why, I mean, one of the case studies that we really talk about, and the first one that we really want to go through was just a class 2 med device in Delaware. I mean, this is one to where they actually had the same cleaning company for years, and they were quote unquote okay with it. But as they started really trying to use it and look at it, they were seeing that they had a lot of particulate, and they were also having lost yield because they were seeing bioburden inside of their product. And this chemical solution the selection that they had, they'd never actually looked to see what it was and the claims that it had on there, any of the disinfectant studies that had to happen. They were not performed. So inside of the actual product contact points, they were using stuff that was not adequate, that was not right. The second side of this one was really actually kind of a larger aspect of this. This was only looking at a focused approach in a small section of it where the product was being adjusted and where it was being formed. Whereas this was, they were having a lot of particulate in the whole space. This was everywhere. From the minute that you went into this non-controlled space, or even this controlled non-classified space, that's where the contamination control program begins. So they were not taking care of the space. They were only looking at that small focus of this, rather than trying to reduce the bioburdens and the particulates in all of the general environment, along with using the proper chemicals and the proper agents when they perform that disinfectant study to be able to go through and to do that.
You know, this is a great point, Brian, you brought up. This is a misunderstanding. Oh, I don't want to do much cleaning or much monitoring in grade D. Why? Everything comes from outside. People bring it from outside. That is the thing that should get the most attention, but the attention is put only around the open product. That is the wrong way of thinking. You hit it at the entry point.
So I think the other one that we talk about in here, and this was a med device up in Massachusetts, and this is an all too common problem out there, is aging facilities that go through retrofits, and it's retrofit after retrofit, and they keep on building on, and they keep on trying to do these things to it. Now, when they do these retrofits, it's really trying to take a look at things. In this specific example up in Massachusetts, they put the equipment built too close to the wall because they were not actually able to get behind the wall and physically remove the bioburdens, the particulates, that they weren't able to get into there. So as they were building on and trying to maximize the space, as every clean room knows, of space is always at a premium inside of a clean room, but there's things that you have to take into consideration. There's the airflow, there's the space behind, there's all of this that goes into the different contamination control program. And the second point that we saw in here was that the cleaning equipment was not right. This was not on wheels, so the people had to pick it up, move it around. Supplies matter. Supplies have a very big effect to make sure that you're choosing the right type of supplies to be able to do that, so that when you're going to be cleaning it into the facility, all of those things into here that goes into that aspect. The last side of it here is it goes back to those good old hippie molds. They're the ones that feed on the cardboard. As they were seeing that there was a lot of organic material that was actually inside of the clean room and allowing for food for the different microorganisms, for things to grow into here. So it was multifold on these types of aspects here.
Yep. So understand your hard surfaces. If you don't understand your hard surfaces, you will not value the importance of supplies. I'm going to ask the whole team a question. Can anybody tell me what surface is image D and what surface is image E? And you can put it in the chat.
Shaylee,
And
are you seeing anybody guess it yet?
Not yet. Come on. Do you have any guesses? Okay, I see a couple.
sponge, glass. Wow. Yes, it is curtains. Mike, Sojourn, and Jim. Good. It is curtains. Curtains are 19th century, guys. Curtains are, they look clean, they look smooth, and Brian and the whole crew of CCS can tell you how difficult they are to clean. Not also because of the nature, but how they are hung. Those are homes for contamination. Your number one, A is your aluminum, B is your stainless steel, and this third C is your epoxy coated wall. Look, now you see why supplies are so important. I'm glad somebody could guess.
I would expect Jim Pollarine to be able to guess that one, so that's always a good thing to see. But yeah, I mean, when we do truly talk on supplies, and another case study that we really had was another med device manufacturer up in the Bay Area that were having contamination issues. Imagine these types of mop heads, and this picture that's here of crumbling, dry, deteriorating, all of those really bad things, trying to get into those clean room curtains, to get into the nooks and crannies, where these were actually leaving all of the particulates inside of the space rather than actually pick it up and take it out of the space. So supplies truly matter. And I'm going to be talking about this for the next, I think, 3 slides, but it really has a very big difference on choosing the right side of this. On top of this, what we saw in this med device company was they were actually having very inadequate cleaning practices. And that was actually not following what they're supposed to be doing on a daily, weekly, monthly type of routine. And it's exactly what Ziva had said before, is truly not taking the consideration, the grade D, the ISO H, the ISO, those outside type of spaces, the controlled non-classified, and including those into those practices. So when you're seeing bad cleaning flow, in this example, they were cleaning from dirty to clean rather than clean to dirty. So they weren't following a lot of this. Now, the main source of contamination was not using the right type of mop heads to be able to pick up the contamination, break the surface tension, use the science behind cleaning was a key thing when we're talking about contamination control, to remove the contamination, not just spread it around. And that's another prime example of spreading it around. And I don't mean to keep on picking in the medical device manufacturers right now, but one of these that we actually saw was the case study was actually a tacky roller was being used on a daily basis on all the facilities on the floors. And the second point of the tacky roller is not being changed out enough. Those with pets and dogs, cats, whatever, they fully understand that you can use a lint roll on yourself, but you can't use it all over without changing it out. It's not going to be picking up the particulates. It's not going to be picking up what you want to do. So whereas the focus at this specific customer was, okay, well, we're going to go and try to save some money by using tacky rollers, because that's a lot less expensive than using the right type of mop head or your disinfectants, whether it's your phenolics or any of those agents inside of that space, was that they wanted to go and try to save money and just pull up the particulate. Where med devices, you have to worry both about the particulate and the bioburdens. And really the frequency of cleanings, they wanted to go and extend it out, but that created a loss of yield of product. So which is better for you, loss of products or doing a right contamination control program from the beginning to be able to have it so that you can get more product out the door.
So let's talk about common errors people make, okay? I tell everybody, don't shift the blame to the disinfectant. This is the best we have. They are pretty good disinfectants in the market, but you have to do your job. You have to know what organisms you have, which are hard to kill and which you should prevent. People bring in pallets, bring in cardboard, and expect the disinfectants to do miracle or the cleaning crew to do miracle for them. Everybody should take responsibility. Disinfectants are not understood. I told you now what disinfectants do and they don't do. They rush to do disinfectant qualification. Yes, it has value, but it's a subjective test, right? You cannot do a test and see. You have to constantly know what is coming in, right? What disinfectants you're using, what supplies you're using, and use the disinfectant qualification data to have a science-based program. Hard surfaces pose a challenge. We talked about it. Supplies play a major role. Clean room design issues you cannot correct them by disinfection. If your airflows are bad, you have dead spaces where the contamination is going in circles. No matter how much disinfection you do, your airflows, your design was not good. Or you have a design where, you know, the pressure differential is not adequate. Every time you open the door, contamination is rushing in. So it's clean. When the door is closed, you open the door, it's unclean. Again, you can't blame the disinfectant. You cannot blame the cleaning. You have to understand the design issues. Procedures not written. Very important that you write the procedures. You manage your disinfectants as GMP materials. Use per manufacturer recommendations. The manufacturers have done all the work for you. You please look at them. If you think you're lacking the data, manufacturers are very open to providing the data on material compatibility. That is something you should do upfront. I have these materials in my facility. This is the disinfectant. Is it compatible with this material or it is going to eat it away? Go ask the manufacturers. Cleaning personnel not trained. I mean, I can talk half a day on all the stuff that I have seen. People using Walmart mop handles, those plastic blue square ones with wipes that are actually little thicker material buffon caps. They have scraped the walls. Because they had mold contamination, they scraped the walls and created more contamination. And then what did they blame? Oh, the sporicidal agent is not working. No, the sporicidal agent is working just fine for the contamination you had. They had deuteromycosis contamination. It works fantastically well. All the sporicidal agents. You have scraped the walls and now you have pulled out all the other molds that were behind the wall in your whatever it's called, gypsum board or whatever it is. Janitorial personnel perform clean room cleaning. I have seen that. I've seen that people actually bring janitorial. I mean, I have a case study. I don't know if I have the time, but I really want to talk about this case study. This is a company I'm going to make it very quick, Brian, if with your permission. This company had mold contamination all over. They called me, they were in the Bay Area. They called me, they were an in vitro diagnostic company. They had fuzzballs in the reagent cuvettes. I went in there. Their filling area was negatively pressured to the bulk manufacturing area. And filling area was nothing but a big room with packaging and forklifts on one side and 10 HEPA filters with curtains around it, opening to the warehouse, right? Fantastic. Who was cleaning it? So they were sending IDs right and left because the sponsor wanted it, and I felt pity for them. And I'm a mycologist. Some of you might know. I said, guys, I will come on the weekend. I will do the IDs. Pay me the hourly rate. Don't spend money per ID. As I'm sitting doing the IDs, I saw a person coming in a Honda Civic. His shirt was, his belly was exposed, and he pulled out a vacuum cleaner. So I followed him, and luckily the entrance to the grade D or ISO 8 area was in front of the lab. So I was watching him. He went in and he pulled out the garbage can and he emptied the bag of the vacuum cleaner, creating a cloud of dust, and then he walked through the cloud of dust. Well, there is a difference between janitorial and clean room cleaning stuff. I'll leave it at that. Hard to clean surfaces ignored, right? Behind, you have incubators and all, you're giving the warmth, you have moisture, and you cannot clean with that behind it. And that's your contamination. Documentation not adequate, and nobody has the time to review.
I think the other one here is that we see a lot there, Ziva, is a triple clean after triple clean after triple clean with the sporicidal agents, but never part of the investigation of where the actual problem comes from, or trying to understand what the organism is. A Band-Aid effect will go there, and it will just be constantly, okay, we're going to do heavy triple clean. Well, there are some major problems with doing triple clean after triple clean after triple clean.
Yep.
So when we really talk about it, and Ziva had hit on there, is there's definitely some areas that get ignored. And this was another biotech company in the Bay Area that had a mold contamination. And honestly, just take a look at the picture. You're seeing that this hard to reach area, what you're talking about underneath of the cleaning space inside of the ISO 5 hood, was never cleaned. Now, partially because they actually never had it in their procedures, and exactly what Ziva talked about the last slide, that they didn't do the documentation, that they didn't say how often you're supposed to be cleaning underneath of this space. Number
Yes.
two, they were only using a 70% IPA solution in there. And if you're paying attention to slide number 2 or 3, 70% IPA will not take care of molds. So they weren't ever taking care of their mold contamination. So this was really twofold, that this is a critical area. This is in an ISO 5 space. And this is what they really had to why they were having their major problems.
Yep.
I mean, one of the other ones that we want to talk about, and everybody who's been in the industry can always remember the good old New England Compounding Pharmacy. And this was a pharmacy that we saw in Southern California. Now, this was on a door hinge outside of the clean room, but this was just a leader and a leading into the spaces inside of the clean room. Inside of this clean room, they were never using any sporicidal agents. They were only using some of your standard cleaners, the standard cleaners that they were going to be using inside of the bathrooms, thinking that, oh yeah, we can use that inside of this space. I mean, partially what we saw in here was an incredibly dirty, dusty, and visible signs of mold, all very, very reminiscent of your New England Compounding Pharmacy. But we knew what the downfall from that being in just the regulations that had to start coming in from this, and the ripple effect that it had across the industries.
I saw the same scenario last week on an audit. All the door hinges were good. One, and they were all consistent, except for one, the door hinges were all across the door and they had issues. Too many hinges and they didn't clean them.
I mean, I think the last one that I really want to talk about here in some of the different contamination sources, like you're ignoring looking at the sources that can potentially create contamination. Incubators, they are fully designed to promote growth of your plates. Well, it can promote growth of other things like your molds. And in this specific example, I mean, this was a company that was using an outside microbiology lab, but this outside microbiology lab, they were using non-sterile water, and they never actually had a PM program set up to shut these down, clean them, and go back and do this. This, once again, where they were starting it down with an IPA wipe and then using a quat. But that's not, once again, the right type of program. That's not following the good procedures, because you're going to have growth of things you don't want. And that's going to just lead to even more problems behind that.
Mold in incubators is a very common phenomena because it has great, conditions for growth. We see this all the time. Let's talk about some dilution errors. Often floors are sticky, residue removal. I just saw a question pop up. How often do you want to remove the residue? Look at how often are you using your residue producing disinfectants, phenolic and quartz. People want to remove residue immediately. You have to take a science-based approach. You want the disinfectant to work, but you don't want a residue buildup. And it all depends on the frequency at which you are going to use the phenolics and quartz. Eyeballing dilutions. The manufacturers have done the work. They're telling you exactly how to dilute it. I mean, I have horror stories. I wish I had a full day for this. You cannot eyeball dilutions. They have to be exact. I'll give you a very good, I don't know if I have the case study. If I have the time, I'm going to give you 2 case studies for eyeballing dilutions or making wrong dilutions. Higher concentration of disinfectant does not mean better kill. Guys, that is good for alcohol, vodka, not for disinfectants, higher concentration. I will give a case study. A client had a mold. It's the Ascomycosis mold. Without understanding where it is coming from, how it came in, they took a sporicidal agent, half the dilution, means double the concentration, and they had cracked walls every time. 2 years later, they were building a brand new facility, and I was on the committee or whatever team for, we were doing contamination control strategy, and they said, oh, we cannot have a peracetic acid chemistry. I told them, guys, you need therapy. You are PTSD to the sporicidal agent, where the sporicidal agent is doing its job. You didn't do your job. You doubled the concentration. You compromised the walls. You created mold. So stop it. Use it at the right dilution. So dilution, please look at the label claim. If you have doubts, the manufacturer is there to help you. So call them. Lower dilution, because we don't like the smell. We don't like the smell of peracetic acid chemistry. Oh, we can dilute it. I've seen contamination issues lost. What is better to do the right dilution than lose millions of dollars of product? Right? Water quality. Yeah, come on. We don't want to buy WFI. It's very, very expensive, very critical. Dependence of janitorial staff to provide supplies. You have to do a good program. You have to evaluate the disinfectants. You have to evaluate the supplies. Using what? using material compatibility data, kill data, and the supplies. Evaluate the supplies. Cleaning is a science. It's not in my past life I did it. I tell people, past life you have reincarnated. This is a new life. Think anew.
I'll run through these kind of quick because I know we're getting a little bit down to the end, a few more things to cover, but this was a dilution error that we really saw. So this was a company that actually used a double bucket system, one that had the solution, one that had the Wi-Fi water in it. Now, when you're going and dipping them back and forth, you're diluting it down. You're having major problems that your solution that you've nicely measured, hopefully, and prepared and documented, you're now diluting that down as you're mixing it together, rather than actually making both solution buckets rather than for that. I mean, the other big problem that we have, and Ziva is probably correct in that we could probably spend a full day of saying these types of problems, but a customer's taking RTU ready to use and diluting it down, either thinking it was concentrate or because they don't like the smell as much. Even though I love that vinegar smell, it's trying to end up, you're diluting down what you've already perfected.
No, what happens is they start with concentrate, okay? That somebody says, I'm going to buy RTU, but they never change their procedure. And they continue. I have so many of these case studies, and it's so silly, you know. It's such a simple thing. Go back to your procedures. What have you done? And then they will spend $150,000 on a disinfectant qualification study, and that diluted down RTU peracetic acid chemistry won't kill stuff, and then they will blame the peracetic acid chemistry instead of looking back at their procedures. What are they doing wrong?
Leading right into your procedures.
Yeah. And the other thing is, this is a lot of audits I see. Somebody likes this disinfectant, somebody likes Fabuloso, it smells good. You can't have, these are GMP materials. There is no centralized documentations. I have seen 6 months, up to 6 months of data fallen down behind the desk because nobody reviewed it. Dependence on contract cleaning company to provide procedures. You know your process. You know you have to sit. You have to figure it out. You don't expect the cleaning company to know your processes, your facility, your surfaces, etc., and your risks. They are not treated as GMP materials. Contract crew is expected to consult. This is something I have seen more often. They ask the contract crew to troubleshoot their contamination issues. That is cleaning companies. They know what they're doing with cleaning. You should know what organisms you have and where they are coming from. It's not the cleaning company's forte. Change I got contamination, I will change my cleaning company. It doesn't work that way. You have to get to the bottom of it. Very soon we will have a bucket train and people will be running around carrying 10 buckets. It doesn't matter how many buckets you have. As Brian said, they were putting water in one bucket and diluting the disinfectant. Wrong preparation will lead to contamination, correct? Preparing stock solutions and keeping in carboy, I told you quartz will grow gram negatives. Alcohols will grow mold and gram negatives. Refilling squirt bottles without ever changing them or that. Well, you will end up in contamination. Fuzzballs. I have seen too many. Please, if you're a cell therapy company, go into your labs, check, ensure that all your IPA is sterile, single use, and you are not refilling.
I mean, I think one of the examples that we talk about, one that we've seen for case study wise, is actually there was a company that was having day shift go and prepare the cleaning solution, leaving it there for the closet for the night stuff, for the night shift. But there were no cleaning logs. There was nothing in there. And I mean, this is another prime example, going back to what Ziva talked about on bleach. It depends on the amount of available chlorine. So what you start at to what you finish at might be very, very different. So this was one of the big problems that we had. I mean, everybody knows in the industry, if there's no solution or there's no documentation logs, it didn't happen. So we didn't know, the cleaning crew did not know whether this was the phenolic of the month, or if it was even the right agent, or if it was the one that they were supposed to be using, the Fabuloso for the bathroom. So some big problems that really can be seen inside of these spaces by just not preparing it correctly.
So documentation errors, contents are inadequate, they don't write, the dilution factor, location of storage not known, procedure review as well, procedure for notification barely exists. Anybody who goes into your clean room should be an auditor. You should look for mold contamination, and I'm going to tell you, being a mycologist and doing hundreds and hundreds of investigations a year, it starts with yellowing of the wall. You start changing color, you have mold growing behind it. So all these things have to be put in place, and you cannot put everything on the exercise of cleaning. It's a whole science, choice, compatibility, residue buildup, supplies, and the cleaning procedure itself. So let's look at Annex 1. Annex 1 is the current regulatory thinking. Why is it important? Right? It is important because all PICS participating countries, including US, participated in doing this, in making this document. You know, it went through from 2017. It's going, we have commented on it. I had a big thesis on this when I commented. Even the convener told me, what, are you writing a PhD thesis? I said I wanted to bring my point home. So what it is saying, the disinfection of clean room is particularly important. They should be clean and disinfected thoroughly in accordance to a written program. We talked about it. For disinfectant to be effective, prior cleaning to remove surface contamination should be performed. Do you see this? You don't clean after the disinfectant, you clean before. More than one type of disinfectant agent should be employed to ensure that they have different modes of action, and the combined usage is effective against all bacteria and fungi. It's good. That means you have a general purpose disinfectant with a surfactant and a sporicidal agent at a minimum. Disinfectant should include periodic use of sporicidal agent, correct? Monitoring should be undertaken regularly in order to assess the effectiveness of the disinfectant program and detect changes in types of microbial flora. How often do you see EM trend report? You talk about excursions. We had more excursions, we had less excursion, but nobody trends the organisms. If you trend the organisms, you know whether you are finding less hard to kill organisms or more hard to kill organisms, and what are the sources, because every trend, whether you do it monthly or quarterly, should have an investigation to understand why are we seeing the changes. Are they seasonal, or is that something we are doing wrong? How can we prevent hard to kill organisms from coming into the critical areas? Cleaning programs should effectively remove disinfectant residues. Nobody is telling you that clean and remove the residue, because you are killing the disinfectant in its infancy. I am telling you for years and years. And there's a study, and that someday I will teach it again. I stopped teaching. I used to teach this course in MIT Endicott House every year for 2 straight days, putting the whole science behind it. Residue buildup should be removed because residue become, but you don't have to remove the residue immediately. You don't. Let the disinfectant do its job. 10-minute, I am not I'm opposed to removing the disinfectant after 10 minutes. You know, I have done studies where I can show you your phenolics as well as your sporocytes can work for anywhere between 2 to 6 hours, depending. It's a very simple study, and I can discuss it or offline with anybody. And that is shows you how long your disinfectants. These people, these manufacturers did make your disinfect for 10 minutes. That 10-minute contact is saying at least minimum, please keep it for 10 minutes, because that is how we got the data. Let's go on.
Yeah, Ziva, so we have ran out of time. So I would love to, if you guys have any other last comments, but we are out of time at this time. So
I think the last one, I think if we could just have just 2 more minutes, Siva, if you want to go through this last slide on here, I think this would be the last part to finish up for EU Annex 1.
They can read this. They're talking about validation studies for effectiveness, but we can have another session on disinfectant qualification and the errors. Let's move on. This is very easy. And it's addressing all the points I said. Let's move on to the next slide. Brian?
So, I mean, only the last one that we have was just where you're talking about disinfectant studies. It's
Yes.
just a subjective.
It's a subjective test, and all this is important. Please make note, when you do a disinfectant qualification study, make sure that whether you or the contract lab have addressed all these key points in their study protocol.
So I think once again, I think we'll kind of wrap it up on this. Really want to thank everybody for the time that you guys have spent listening to both Ziva and I. I know that we unfortunately don't think we were able to get to many of the questions that were in the chat here, but Ziva and I will definitely reach out to each and every one of those. But there's the contact information for both Ziva and myself, and we really look forward to hearing from you guys and working together. I mean, Ziva, did you have anything else to add there?
No, we will answer all the questions because these are burning questions and there's a lot of misinformation.
Okay, great. Shaylee, was there anything else closing there?
nope, other than this presentation will be provided to everyone. It will be added to the link that you joined the webinar on. So please look out for that. And also this recording will be available to you afterwards as well.
Great. Thank you,
All right.
everybody.
Well, great. Thank you. Have a great rest of the week.